So, good morning, everyone. I'm Shivansh from KS Medical Academy, and I'll be speaking on the topic Vibrio Cholera. Vibrio Cholera, as we all know, belongs to the group A species of Vibrios. So, these species, the Vibrio species, they are gram-negative, monotrichous, and curve-shaped bacilli, requiring a pH of 8.2 to 8.4, that is alkaline pH for growing. The special thing about Vibrio Cholera is it does not need salt concentration to grow. That is, it is the major exception because all the Vibrio species except Vibrio cholera and Vibrio mimicus, they require salt concentration. So Vibrio cholera species, they don't require salt concentration, they can grow without salt. Coming to the biochemical test, so first test that we look upon is oxidase test. Oxidase test is positive in case of Vibrio cholera. Catalase test may be positive or may be negative. Indole test is positive. Nitrate reduction test is positive, so it would convert nitrates to nitrites. These nitrites would join with the indole, that is formed by breakdown of tryptophan. This also tryptophan would be broken down into indole by Vibrio cholera. So the indole and the nitrate, they combine to form nitrosoindoles. Nitrosoindoles, when mixed with acid, they give us a red color. Thus cholera red test, that is the specific test for Vibrio cholera that we do. Then we can do another test, that is the gelatin test. So gelatin stab test, what we do is we'll get a napiform liquefaction in that. Then in slant medium, what it would do is that it would produce acid and gas, but the gas is not H2S. Now, what specimens we collect in Vibrio cholera? The specimen that would be collected in Vibrio cholera is the stool samples. So the stool samples are needed to be transported to the laboratory. So we can use three mediums. One is the VR medium. Second is the autoclaved seawater. The third one is the Kerry Blair medium. So, as Vibrio cholera is a non-fastidious organism, so the non-fastidious organisms, they grow on simple medias. So for that, we'll for the enrichment media, we'll be using alkaline peptone water. Along with that, we can use monsoon staurocolate tellurite peptone water. Tellurite peptone water. So these two are the enrichment media that we use. Okay. So then we'll have a specific media, that is we'll grow it on TCBS agar. So TCBS agar, it would give yellow color along its colonies due to the sucrose fermentation that these species show. Then in blood agar, they would show the hemodigestion. So this is a characteristic property, and we need to test it for Vibrio cholera. So it would help us in diagnosing that the cholera is formed by Vibrio cholera. Then we have the conky agar, that would test for lactose fermentation or not. So these are non-lactose fermenting, or we can say these are late lactose fermenting, as they would start fermenting after 2 days. Then the main important thing is the division. The Vibrio cholera, they are divided into O1 to O100, O200. So this is O is the antigen that we are using to divide them, like, on the basis of antigen. So group A and group Vibrios were divided into group A and group B based on H flagella antigen. In group A, we had Vibrio cholera. That is the only species that come under group A. So Vibrio cholera is further divided into O1 to O200 based on the O antigen. So O1 and O139 among them are the most important because these contain the cholera toxin, and they are responsible for forming cholera. So among the O1 and O139, the most important is O1, as this is the main culprit for causing cholera. So cholera, when it is an endemic, it is only restricted to children. But in an epidemic state, that the cholera epidemic, there have been seven. We are currently in the seventh epidemic. So in these seven epidemics, they did not involve not only the children but also all age groups. So epidemic wise, it would not see the age group, it would be spread among all the age groups. Then coming to the another important point, that is the pathogenicity factor. So the pathogenicity factor is important because these would help them to establish their colonies in our bodies. So the first thing is, it, as I told, these are monotrichous. So they have a flagella. So the flagella is sheathed. So this, what would this sheeted flagella would do? It would help them to enter the intestine of the host. Then once they have entered, they need to stick to the surface. So they need pili. So what would happen is they have toxin co-regulated pili. These would help in attachment to the surface. Then coming to the main toxin, that is the cholera toxin. So cholera toxin has a special shape, we can say. So that is AB5. So it would have one A subunit and four subunits of five subunits of B surrounding the A. What happens is the cholera toxin, it is a CTX5 phage mediated toxin, and this is the most important thing that the Vibrio cholera would possess. The O1 and the O139, these are the two subspecies that contain these things, this toxin, thus causing cholera. The main receptor that the cholera toxin it binds to is G1 ganglioside. So what happens is now I'll tell you about the how it acts. The cholera toxin acts. So the cholera toxin it would go and bind to the GM1 ganglioside receptor. Once it binds to a receptor, it would be internalized. That is endocytosis takes place. It would go to the endoplasmic reticulum. In the endoplasmic reticulum, what happens is the A subunit splits off. A subunit divides into A1 and A2, and the A1 is the major, that is the active portion that would cause the phosphorylation of the CFTR. So how this phosphorylation is caused? It would, the A1 subtype increases the CAMP concentration, CAMP, cyclic AMP concentration, that would increase CFTR, that would further increase the secretions of the intestine, thus causing watery diarrhea. That is a characteristic feature of cholera, Vibrio cholera, which is caused by Vibrio cholera. Coming to the next factor is, that is the neuraminidase. Neuraminidase, what happens is, for cholera toxin to bind, we need the receptor. So GM1 ganglioside receptors are formed by neuraminidase. The next pathogenic antigens would be SORT and the ACE. So these play a minor role in causing cholera. And the last three are the collagenase, mucinase, and elastase. Okay, so coming to the cholera, I'll just give a brief summary. So cholera is basically only human. It is restricted to only human population, and as I told, endemic to children. And when an epidemic hits, it is spread in all the age groups. So what happens is the minimum concentration that is required for a bacteria to cause infection. In this case, the Vibrio cholera, they require, as I told, the Vibrio species, they require pH of 8.2 to 8.4 to grow. So these cannot grow in acidic medium. So a high load is required when we ingest these as it is. Like when we are having, when we get it infected, like without having any food or any of the HCl blockers. So what happens is when we are having food, it would alkalinize the pH. That would help the bacteria to colonize even at lower concentrations. Then coming to the next part is, the next thing is, there have been six epidemics and the seventh one is currently going on for the cholera. And for cholera, as I told, gram staining would be negative, gram negative, monotrichous and curve shape bacilli would be seen, and the rest of the test would be same what we will be conducting here. Serogrouping based on the O antigen, O1 to O200, O1, O139 important. O1 is further divided into LTOR and classical part. So among them, classical is the most dangerous, but the most number of carriers are produced by the LTOR part, LTOR subgroup. Then toxicity test. It is basically we need to have cell line assay that we do. So cell line assay, it is done on Y1 adrenal tumor cell lines and Chinese hamster ovary cell lines. Now coming to the treatment of part of cholera. So now firstly cholera, as it produces a lot of water loss. So first primary motive would be to regain the water loss, regain the water and the electrolytes. So we are going for rehydration. Rehydration we'll use ORS therapy. Based on the amount of water that is lost, it can be intravenous or it can be oral. Then coming to the next thing is antibiotics that can be given. So the drug of choice that we use is azithromycin, that is given only one dose. The rest drugs, doxycycline can be used. We can also use erythromycin and ciprofloxacin. There are vaccines that have been produced for cholera. So these are vaccines would be of two types. One is the attenuated, live attenuated, and one is the killed. So live attenuated, we have orocol. So orocol can be given only in age groups more than 18. Whereas the killed vaccine, that is a decoral, that can be given above the age of 2 years, and it contains the Vibrio Cholera O1 species, O1 non-toxic, which is non-toxic part, and a non-toxic B subunit Cholera toxin. With this, I end my topic. Thank you.